Hello, it's a great pleasure to spend today's hour with you. I want to express my gratitude to the International Toward Society for giving me the opportunity to share our work. And I also want to thank specifically Professor Sloboda and Professor Matthews for nominating me to be the Canadian contingent for this exciting lecture series. So today I'd like to share with you our work on epigenetic embedding of early life experiences, and really trying to understand how early life environments get under the skin to affect health and behavior across the lifespan. Before I do this, I would like to acknowledge that the BC Children's Hospital Research Institute, where all of this work has been done, operates on the traditional, ancestral, and unceded territory of the Coast Salish peoples, the Musqueam, the Squamish, and the Tsleil-Waututh nations. Further, this acknowledgement, gratitude, and respect extends to all of the First Nations communities on whose traditional territories the Research Institute builds relationships and operates in British Columbia. Now, what I'd like to do today are basically three things. I'd like to discuss with you the biological vestiges of early life environments, describing our society to cell paradigm. I would also like to spend a little bit of time to discuss some of the challenges that we face when we do this work and some of the opportunities that arise from it. And lastly, I would like to close off with the idea that there might be an epigenetic clock that we can explore to understand children's health better or from a different perspective. With that, collectively, what I hope to do is to share with you why we think that epigenetics is really one of the key players at the interface of nature and nurture. Now, before I do this, I also would like to acknowledge that this entire piece of work and research that I'm going to show you has really been done by a wonderful and amazing and outstanding set of trainees, and staff, and mentees, and a fantastic set of collaborators. And I'll try my best to highlight everyone as we go along. Now, you might have heard of epigenetics. There are many definitions that are being used around and changing. Perhaps the most commonly used, you can see here on the top of the slide. Our particular aspect that we're really interested in is really more focused on the social part of it, and that is, is epigenetics a process by which life experiences influence the way our genes are expressed? And we do this really by a very simple paradigm. We postulated there are certain experiences or environments that get under the skin to affect biological pathways and drill all the way down into the nucleus of the cell to affect and alter the expression of genes, and doing so sometimes in a persistent way. Of course, this is not a one-way street. You can easily imagine it going the other way as well, as outlined here. But today we're going to mostly focus on the left hand of this paradigm of these pathways. Now, if we want to understand epigenetics, we need to understand that the DNA that is shown here at the bottom doesn't really occur in the cell as a naked entity. Rather, it's packaged into multiple and very complex patterns around different proteins, so-called histones. And so there's really a very complex landscape of epigenetic marks and mechanisms. Each of those really has a terrific story to tell. And my former postdoc, Maria Ares-Di-Sabal, has actually summarized this and shown in this figure here in a paper in PNAS in 2019 that I highly recommend you to read. So today, though, we're going to focus primarily on, or exclusively on, DNA modifications, and they're really only on cytosine methylation. So the most canonical of these DNA modifications. And in human population studies, which is what our work is entirely based upon, these are, I think, at least 99% of all studies are done focusing on DNA methylation. And so this is going to be our workhorse for the rest of this talk, but I do encourage you to think about all these other modifications, and for the students amongst you to imagine how all of these other modifications could be explored as it relates to this idea that epigenetics is in part an interface of nature and nurture. Now, I tend to think very simply. So my view of epigenetics is really shown here. And what I like to often think is of a gene basically being a light bulb. And you can imagine a light bulb being either totally on, like on the left side, or totally off, like on the right side. Or if you live in a fancy house and you have a dimmer, the light bulb might be anywhere in between, depending on how the dimmer is set. And so really, if we think about DNA methylation, specifically in gene regulatory promoter regions, we can just simply think of DNA methylation serving as the dimmer and the mRNA, the gene expression, being the light bulb. And so if we think about this, pick your favorite gene. Your favorite gene can be is now totally on. The dimmer is all the way up, and on the left side of the piece of DNA, there is no methylation. Now we start methylating this DNA. You see there's a methyl group on top that's attached to the DNA covalently. It pushes the dimmer down. Second one comes in, pushes the dimmer further down, and the third one comes in, pushes the dimmer all the way down, and our gene, our light bulb, is turned off. So that's really all you need to understand if you want to understand the basics of human population epigenetics. And for the talk today, whenever you see a dimmer, it really means that what we're interrogating here is CpG, or DNA methylation, in particular regions of the human genome, using array technology that we have been using for the last 10 years or so. And when you see a light bulb, it means that it indicates that we measured the mRNA. Now, what kind of changes can occur? What kind of forces or drivers exist to DNA methylation? First of all, of course, tissue specificity, and we come back to this later. Each cell or each tissue of our body has a distinct DNA methylation pattern, but then there's also environmental influences that associate with changes in these DNA methylation patterns. And first and foremost, population studies and looked at historical occurrences such as the Dutch famine, work by many different people, but including Bas Heijmans, has shown that the nutrition and the lifestyle of the mother during pregnancy affects the epigenome of the child, and this has been replicated many times and for many different prenatal exposures, including starvation, cigarette smoking, and the like. Work really pioneered by Michael Meany and his team at McGill University in Canada, and now followed by many different groups, including Professor Matthews, has shown that the maternal care associated with the epigenome of the child, in many cases, doing so in a lasting fashion and in genes relevant to things like stress response. And as you can imagine, it's this kind of work that really drives the importance and the excitement of epigenetics into the public space as well. Lastly, most of you, of course, know twins, identical twins, and so they, of course, basically have the same genome, save for some changes in the immune system. But what you might see here is like these two twins with, cutie on the right being all content and happy, and the one on the left crying and being upset. And so work by many different groups, including most recently Jonathan Mill and Art Petronis and others, Manale Stella, has shown that identical twins initially have very concordant epigenomes, DNA methylome, so that dimmer switches are very similar. But as they go through life, they become more discordant, and especially in cases where they happen to be separated early in life. Again, highlighting the importance of this early life embedding of epigenetic mechanisms. In turn, these differences, even in monozygotic twins, can be associated with differences in phenotypic outcomes, including many different health and disease risks. So collectively, I think all of this shows that epigenetics is really potentially the interface of nature and nurture, and this is really where our lab is situated in. So the nature on the left is the blue, is the DNA. The nurture is this sort of fertile ground on the right, with family environments and the like. And we postulate that epigenetics, together with genetics and the environment, interacts to basically ensure that our children all get a healthy start to life. And what we're trying to understand is how can we explore the science to prevent diseases that are rooted in early childhood. And our lab has been doing this very much on a developmental context. This is taken from a great review from a postdoc, Megan Jones, wrote a couple of years ago, where basically postulated, again, very consistent what we know from child development, broadly speaking, that there are sensitive periods, and what we show here in this top figure is that if an exposure happens during a sensitive period, and say this is a detrimental exposure like child abuse or any of these kind of things, it might alter the trajectory of the epigenome in this red line to lead to a higher risk for a given individual for detrimental health outcomes. However, if the same exposure happens in a later period, when the sensitive period might not be as receptive, if you wish, for the same exposure in the epigenome, it doesn't necessarily alter the trajectory of these the epigenome to an extent that results in reduced normative risk. Of course, this is much more complicated. One can think of the sensitive periods later, not just during gestation and early life, but for example, during puberty and the like. And we're currently exploring all of this. But the general message that I want to send here is that it is very clear that the epigenome does associate with changes, and that we need to look at this in a developmental context. And my mentor and hero, the late Clyde Hertzman, very consistent, of course, with the developmental health, developmental origins of health and disease hypothesis, has put this into a beautiful concept, which he calls the biological embedding of early experiences and how this affects health in adulthood. It's in my mind a classic read that I encourage everyone to have a look at. Now, we have been really standing on the shoulders of giants to explore whether indeed epigenetics is at the interface of nature and nurture, and whether epigenetics indeed can serve to associate or associates with a number of environments and experience that we know from epidemiological studies and from more directed studies have an effect on child health and well-being. And so with a wonderful set of collaborators, many of whom are listed at the bottom here, and as I said, my amazing crew of staff, mentees, and trainees, we have set out to test in various paradigms whether early life experiences get under the skin through mechanisms associated with epigenetics. And we've been focusing originally, and we'll talk about this in the next few slides, primarily on early life socioeconomic status. As you might know, early life or socioeconomic status is generally associated with poorer outcomes, health outcomes, cognitive outcomes, developmental outcomes. And interestingly, if this happens during the first few years of life, it often sticks with us for the rest of our lifetime, irrespective of our future socioeconomic status as adults. Furthermore, we also have been focusing on early life parental stress. Does it matter whether you grow up in a family with doggy woof woof, nuclear family, or in a family where the parents constantly yell at each other? And ultimately, how does that relate to childhood temperament, behavior, and trajectories? And so today we're going to touch on all of those aspects using selected highlights from our studies that we've done over the years. Now, historically, we have been starting off with looking at socioeconomic status early in life, and this is work we published with Greg Miller and Edith Chen and their team already in 2009 in PNAS. But I want to highlight it here again because it, I think, sets the foundation for much of what we have done since. And so in this study, we had a group, a cohort of adults that was distinguished by whether they grew up in low or high socioeconomic status during the first 5 years of life. And they were then mixed, happened to be mixed, or they were selected to be mixed in adult socioeconomic status, so that whatever we were looking at would really be not confounded heavily by adult socioeconomic status, but could be traced back to childhood socioeconomic status. And so we did a number of things. First of all, at the time, we didn't yet have the technology to look at the dimmer switches, so we focused on the gene expression patterns in these white blood cells from these folks. And what we found is that there was indeed a difference in a number of several hundred genes in terms of their expression pattern between folks who grew up in low early life socioeconomic status versus high socioeconomic status during early life. This was not a random pattern. This actually was a pattern that suggested had particular enrichments for transcription factors involved in inflammation. It suggested that folks who grew up in low socioeconomic status had a pro-inflammatory pattern in their blood. And in fact, if you look at the right top, when we then test this directly by stimulating various innate pathways, the toll-like receptor pathways, innate immune pathways, we do find consistently that the white blood cells from folks who grew up in low socioeconomic status have a stronger response when challenged with these TLR agonists. Further, at a whole body level, we find that folks who grew up in low early life socioeconomic status have a higher output during the day of the stress hormone cortisol. And so really showing that along from the very genes to the immune function to the physiological response, there is a biological residue of low early life social class. Now, interestingly, in work that used the same cohort, we found that buffering and resilience by maternal warmth can actually dampen some of these effects, and so parents can make a difference. Several years later, the technology to measure these dimmer switches became available. And so in another paper in PNAS, we in fact then tested to the extent that there was an association of DNA methylation with early life socioeconomic status using the very same cohort. And what we found is that as judged by these by the right skewing of the p-value distribution on the left, there was in fact an association of DNA methylation with early life socioeconomic status in early life. But interestingly, if you look at the flat p-value distributions on the right, there was at least in this cohort under these conditions and with this technology that we applied, no or very little association of current SES. So collectively, this suggests that early life SES associates with distinct DNA methylation patterns in white blood cells, that it associates with a pro-inflammatory gene expression pattern and increased inflammatory innate immune response, and as well increased cortisol. Of course, this was the first study, and so it became really important to then test to what extent these things would replicate. And further, of course, as you can imagine, socioeconomic status is a very complicated construct. And so in the next few slides, I will show you again examples from studies where we attempted to test whether this could be replicated, and perhaps also whether we could see this in a bit more granular way. So in recent work with Tom Boyce and Nikki Bush, both from the University of California in San Francisco, using their PAUSE cohort from the East Bay and East Bay, Berkeley, and Oakland. What we tested here is whether three distinct components that make up, in part, socioeconomic status, those being income-dependent, family adversity, and parental education, do associate as well with DNA methylation, in this case in cheek cells. And as you can see by, again, these right skewed p-value distributions for all three of those variables, but also at the bottom by these volcano plots, which are basically visualizations that allow you to test or to visualize each CPG separately, and the higher up on the y-axis a CPG is, the more likely, the more statistically relevant it is, and the further it goes away from the middle, from zero, the more biologically, the stronger, larger biological effect size it has. And so what you're looking for are basically these blue and red dots for all of these associations. And when we quantify those, what we found is that all three of those variables indeed do have a fair number of CPGs associated with them in these buccal cells of these 8 to 10-year-old children. Although the numbers differ from variable to variable, and there is some overlap between the variables, but perhaps less than we were initially expecting, given that they tend to be highly correlated in many ways. So again, this shows that in an entirely different cohort, in an entirely different tissue, this being buccal cells, and in an entirely different age, this being children versus adults from the previous study, we can see associations of components that make up the early life environments with children's DNA methylation. Now, together with Tom McDade and Chris Kusawa, who many of you will, of course, know very well, we're also testing this, whether this could actually be seen in places outside of North America. And so Tom and Chris have long been working in the Philippines with the Cebu longitudinal study. And here, again, in a paper that was published just a year ago, what we found is when we tested, again, various components that make up socioeconomic status during childhood and whether they can be associated with DNA methylation in young adulthood, we again found many CPGs, for example, on the left here, those that associate with folks who were for the life course in low socioeconomic status versus high socioeconomic status, but then also more granular patterns related to household assets, income, and education. So very similar to what I showed you from the past cohort. Again, suggesting that this can be actually seen irrespective of what cultural or background we're looking at. In work that then extended this a bit earlier with Tom and Chris, we actually also looked at a broader range of social and physical environments early in life, and we found that things like exposure to animal feces, being born during the dry season, or breastfeeding, all, as shown by these volcano plots, all have some statistically significant association with a small set of CBGs. In this case, the study was focusing exclusively on inflammatory genes. We also then calculated an inflammation index based on the markers that Tom and Chris had collected and associated with various DNA methylation sites. And of course, the hope was that the sites that were associating with some of these environments would be this, in part, at least the same as the ones that gave rise to the inflammation index. Unfortunately, that was not the case, so there's much more work to be done. Now, this was just one really a few examples that focus on one of the aspects that we are looking at in our laboratory with our collaborators, that being socioeconomic status during early life. There are a number of other papers that we've published that sort of expand this. We've shown with Marilyn Essex in Wisconsin, for example, that there is a signature of childhood stress and on DNA methylation in adolescents. With Megan Gunner, we have found that we can see distinct DNA methylation patterns, but also differences in blood cell types in adolescents that spent the first 2 years in orphanages in Eastern Europe. And with Andrea Roberts, more recently, my PhD student Nicole Gladish has found that we can see signatures associated with childhood abuse in male sperm. So just to give you a bit of a flavor where we're taking this, and I want to spend the next few slides showing you some work that we're really taking this very much into the family unit, trying to understand, epigenetic associations, whether they exist and how in turn they might be associated with child behavior. And so this work started with my former postdoc, Sarah Moore, who actually drew upon a cohort that has been established in Vancouver, the time of interaction between an infant caregiver and the infant was measured for an entire week. You can see here on the left that there's quite a range of caregiving time that an infant spends with a given caregiver, ranging from a few hours a day to almost 22 hours a day. And this is, of course, in some extent reminiscent of the work that Michael Meany has been doing with his rat licking and grooming paradigm so beautifully. Now, what Sarah did in this study, she tested again in buccal cells whether she found associations, whether there were associations with given CpGs, and in fact there were a number of those that she could identify, as shown on the right here, including in some genes that might have relevance to immune function and the like. And of course, again, this is the kind of work that gets you into the newspapers, and so it's just to have for fun a headline from one of the national newspapers here in Canada that we was featured this work. More recently, we have been working with the APRIN cohort in Calgary, in Alberta, primarily Nicolas Tourneau and Jerry Giesbrecht, who many of you will again know, and a wonderful postdoc, Sarah Merrill, has really merged her interest in infant attachment with our expertise in DNA methylation in the context of this cohort. And in a paper that was just published a couple of weeks ago, what Sarah did, she tested whether there was a methylation difference between children that had a secure attachment style. So these are the children on the left with a B versus children with an insecure attachment style. And for this, she grouped children with anxious, disorganized, or avoidant attachment styles. And again, as you can see on the bottom left here, in this volcano plot, there was an association. She identified a number of CpGs with a fairly rigorous statistical threshold to be different between children with secure versus insecure attachment style, and this is done in white blood cells. Importantly, and I touched upon this earlier already very briefly, what we can also do is we can use these DNA methylation patterns to actually bioinformatically derive the underlying cell type differences based on the fact that each of these white blood cells has a distinct methylation pattern that can be basically exploited to give to have a very good measure of individual differences in blood cell types if, for example, as in many cases, there is no complete blood count available. And so what Sarah found, this is part of our general approach to use these methylation data. What Sarah then found here is that the children with the secure attachment style actually had higher proportions of the monocytes in their blood than the other the insecure children, and really fascinating because this was consistent with some older data that were in the literature, and so we're exploring this further. But I wanted to show it to you because it goes to show that these DNA methylation patterns can be explored and exploited in ways beyond just looking at individual CpGs. They can also serve as a surrogate for differences in cell type or for cell type measures, and we'll touch at the end of the talk how they can also tell us about epigenetic aging. Using the same cohort, Sarah has also then looked a little bit earlier, and so what she tested here in this paper that was published, I think, this week, she looked at whether there was an association, as many had predicted, between paternal ACEs or parental ACEs. In this case, she looked at paternal ACEs and DNA methylation in the offspring, and indeed, she found a handful of CpGs that were associated with the total paternal ACEs in these the blood of these children at 3 months of age. She then also used more deeper analysis to see whether to the extent that these CpGs were also associated with other things, because they had been, the same CpGs had been actually found to be associated with sleep and ADHD and so on. And the bottom figure, what you can see here is that there were actually a number of CpGs that were both correlated and highly statistically between not only the paternal ACEs, so before birth, but also infant sleep latency at 3 months, and even child and attention hyperactivity at 3 years. It's a bit of a complicated figure, and I strongly encourage you to look at the paper. It's really fascinating because it goes to show how we can, moving forward, go from these simple association studies into more sophisticated studies as more and more data became available, and studies and CpGs can be cross-referenced between different data sets. Now, I'll just spend a few minutes on some of the challenges and the opportunities that we have, and really just focusing on two. One of them being the tissue specificity that we already touched upon in the context of the cell type differences, and the other one, the fact that contrary to the very early work, we believe there's much more of a underlying effect or relation between genetics and epigenetics. And so, as I mentioned, each cell, each tissue, each cell type has their distinct metallome. And for us, as you can tell, a lot of the work that we do really has to do with child behavior. And so what you would ideally want is to look at the brain. Of course, that's impossible to do in living people. And so what we need to understand is, to the extent that measures in peripheral tissues such as blood or buccal cells do indeed report on the brain. And so some years ago, we teamed up with Gustavo Turecki and Michael Meany from McGill University, and this was work done by a wonderful former bioinformatician, Rachel Edgar, who's now doing her PhD in Cambridge, where we used much postmortem tissues to really get a flavor of what are the differences between blood and brain. And if you look at these, the two volcano plots in the center of the slide, you can see that the left one is blood versus one brain region. You can see a really very robust differences versus if we look at two brain regions against each other on the right, these are much less. So clearly blood is quite different. If you look at the straight up correlation numbers, they're actually quite high still, even between blood and brain is 0.89, as you can see on the left here. I think this is in part because many CBGs tend to be invariable that are being measured with this technology. But nonetheless, really blood is quite different from brain, as you perhaps might expect. Now, Rachel really did something wonderful. She put a Shiny app together where anyone who has a blood methylation pattern can actually query their CBG. And so what you'll find is that there, as you can see on the right, there are in fact, a number of positively correlated CBGs, where basically whatever you measure in blood reports almost precisely on the DNA methylation pattern in brain. There are also negatively correlated CBGs, like you can see on the left, and I would argue those are also very informative. But then there's a fair number of these uncorrelated CBGs, and those are probably the ones that show up in the volcano plot. So I encourage everyone to use this tool. It really helps to at least get a flavor to the extent that something you measure in blood does report on the brain, for those of you who are interested in brain DNA methylation. Similar to very talented grad students, Sumaya Islam and Sarah Goodman have built on this idea that there are differences between tissues, looking at matched tissue samples from blood and buccal cells, but I did that also now adding into genetics into the mix. And this is a complicated slide, and I don't want to go too deep into it, but sufficient to say that if you look at the lower half of the left side, you see some of what are called these MQTLs. And so you can see at the bottom, there are 3 different genotypes, and on the y-axis, there is the associated DNA methylation pattern. You can see beautifully on the left 2 boxes that for this particular CBG, there are these MQTLs are the same between buccal and blood. If you move to the right, you can see there is a MQTL, a CBG that's only has an MQTL and PBMC, because there is no differences between the different genotypes in buccal. And conversely, the bottom one is one where you have a MQTL in buccal, but not in PBMC. So very complicated. So there are certainly many CPGs that are under some genetic influence, but then they can be tissue specific, but don't have to be tissue specific. And so Sarah and Sumaya set out to catalog those, and then to as you can see on the right side of the slide, to look at existing studies, to what extent they reported on different CPGs belonging to different categories in their cataloging. That is, cataloging is either an MQTL an informative side, as they call it, or differential side. And informative and differential simply refers to the tissue that they're reporting on. And you can see that just in these 5 exemplar studies of childhood DNA methylation studies, that there are pretty dramatic differences as to what category these different studies report on. And we're trying to understand what those are and how this could be used to perhaps inform future epigenetic studies further. So stay tuned. I think this is one of the most exciting areas to explore the interface between genetics and epigenetics. So just to finish up, what I've shown you so far is really epigenetic associations. And they're wonderful studies because they identify specific CpGs that are associated with a particular phenotype, with a particular variable, or particular CpG, pathways like inflammation pathway. They are, as we touched upon, often tissue specific, and so we need to be careful as to what we learn from a given measurement if we want to extrapolate to different tissues. And as these technologies become more complicated and we measure more and more CpGs, I mean, we in theory can measure all 28 million nowadays, and once we figure in even genetics, the statistical difficulty becomes quite challenging. Now, Steve Howard some years ago, based on the fact that many of these epigenetic marks also change during normative aging, realized that perhaps we can actually create something like an epigenetic clock, and Steve and many others have done that so beautifully. And so basically, the idea here is that it identifies age, if you wish, and depending on how the clock is trained, it can be fairly tissue agnostic, and statistical difficulty tends to be fairly low because all you need to calculate is the difference between your predicted epigenetic age and your chronological age. And again, my former postdoc, Meg Jones, who's now in Winnipeg, put this so beautifully in a review in Aging Cell a number of years ago, where basically Meg postulated that if we look at most people, when we compare their chronological and their predicted epigenetic age, they will be in the concordance zone. But then there might be some folks who, are epigenetically old, or folks like Professor Matthews, who might be chronologically 50, but epigenetically, say 40, would be epigenetically young. And so this then translates into differences in relative health, where, for example, folks who are epigenetically old tend to have shorter and more detrimental lifespans compared to the concordant folks and the folks who are epigenetically young. And many studies have since shown that is indeed the case. Many different health outcomes, many different variables, including some social variables, are in fact associated with deviations between chronological and epigenetic age. And I just want to show you one that we have been working on again with Greg Miller and Edith Chen, coming back to where we started, these cohorts that separate, that stratify, look at the effects, the lasting effects of low early life socioeconomic status versus high socioeconomic status. And what we've shown with Greg and Edith in a paper published in 2018 in Psychoneuroendocrinology was that in fact, folks who grew up the first 5 years in low socioeconomic status tend to be epigenetically statistically significant older than folks who grew up in high socioeconomic status. And again, this is largely irrespective of current socioeconomic status in these DNA methylation, DNA aging measures in purified monocytes in this particular case. So really fascinating, again, using a different information content of the epigenome to test the effects of early life environments and experiences in lifelong health. Now, as we've done many of these studies, we realized that particularly for children, the noise for these studies tends to be quite high because it's around plus minus three years, and for a 70-year-old that's not a big deal, but for a 4-year-old it is. And so we then teamed up with Steve Horvath, an extremely talented and very successful former PhD student, Lisa McEwen, took advantage of the richness of data that we had in the pediatric range in our own laboratory, in particular in buccal cells, to create a new and improved epigenetic clocks for the pediatric age range in a paper published a year and a half ago in PNAS. And what Lisa did using elastic net regression, and she basically came up with something like 90 or so CPGs that very accurately predict, especially in younger children, their epigenetic age and how it correlates in buccal cells and how it correlates with chronological age. So we're now actively exploring this, and just to give you a little bit of a flavor, in this paper, what Lisa, together with Kieran O'Donnell from Michael Meany's lab, found is that there is actually an association of gestational age with the predicted methylation age. Also using a publicly available data set from John Crelly's lab, we found that there were deviations between children with autism spectrum disorder on the left and normative children on the right. Again, this was very early work, but I think it's very instructive and gives you a flavor of where we want to go with this. And in fact, we now have a very robust program in place that helps us to put the PET-PE clock into context. We want to understand what are intrinsic factors such as sex, gender, age, the genetic underpinning, and what are the physical and social environments, prenatal or postnatal, including many of the ones that we talked about, but not limited to those. How do they associate and drive the pediatric clock? And then in turn, how is that affecting, how is that reporting on health and developmental outcomes? And of course, we also would like to test whether some interventions then do affect some of these associations. And again, I'd love for any of you to become a partner in this journey. If you have any cohort where you collect buccal cells that you would like to work with us and partner with us, please contact me at my email shown here. We very much look forward to really understanding the pediatric buccal epithelial clock and putting into context. Now, to summarize, I hope what I shared with you today is some of our excitement that positions the epigenome at the interface between early life experiences, environments, and later life phenotypes. We touched upon how this potentially needs to be looked at in the context of tissue specificity, and of course in using a developmental lens. And we very briefly touched upon how some of the genetic underpinnings might affect all of these associations that we see. So I just want to point out that there are many different experiences, environments that associate with DNA methylation, and in turn, DNA methylation associates with many different phenotypes, but none of these, by our current study design, allows us to invoke causality. So it's really correlations at this point. And of course, we look forward moving beyond correlations into causality studies. So with that, I leave it. Is it really nature, DNA weighing down the scale, or nurture, or both? Thank you so very much, and I look forward to the discussion. Thank you
very much, Mike. That was just an outstanding presentation, and I apologize for the technical issues at the beginning, but I think that talk was just fantastic. So we have some time for questions, not an enormous amount. There are a couple of questions that have already come into the chat, and I'm going to just combine them. Shahiba Mohammed and Victoria Lima Rogel really asked similar kind of questions, and that is essentially, you're seeing a lot of the impact around early environments, and then of course now with the current situations around the world with instability and war, what are the potential impact of those environments on the epigenome?
Yeah, those are great questions, and very timely, of course, and in many ways really dealing with some of the issues that we're facing as a worldwide community. So I don't have the answer just yet, but I can tell you that just last week we got 400 buccal swabs from Syrian and Jordanian refugees in collaboration with Professor Rana Dajani. And we'll be looking at both epigenetic wide association studies as well as the pediatric buccal clock data in that cohort that also actually underwent an intervention in some of the refugee camps. Connie Mulligan from Florida already has looked in similar population with on some candidate genes and found some interesting associations. So I think we'll certainly see some signal given that, you know, the amount of trauma and the amount of stress that these children and adolescents experience there is traumatic from, you know, what I guess. But at the same time, perhaps also because they are resilient children and adolescents, perhaps we can start, you know, developing some understanding of whether there are signatures of resilience that could provide some more hope than the usual sort of like, oh, it's all, you know, detrimental and bad. So,
yeah, I think that's a really important point, isn't it? Because when people talk about epigenetics, they're normally talking about negative things impacting the methylation and other epigenetic readouts. But in fact, epigenetics probably plays a really important way of understanding resilience, and it's an area that needs, I think, a little bit more emphasis in the field.
Yeah, it's harder to get funded, but I totally agree with you, Steve. It's of course, you know, if there's some positive messaging that can come out of this work, I think it will be good for everyone involved.
And then, sorry, Lucilla.
I was just going to add, thank you very much, fantastic talk, and I love the comparison of the cells. I've done epigenetics work myself in cord blood, and we always worry about whether it relates to the brain or not, but that's really, really super parallels there. I've never seen it before. Fantastic, thank you so much. Just a quick question for me is that stress during these difficult times with either in Syria and Afghanistan and so on, or indeed in any situation with bondage with a child, also is associated with changes in nutritional status, in particularly breastfeeding difficulties, you know, with young children and so on, and then in war-torn countries with absence of food and so on. So, I mean, obviously your predominant hypothesis is stress, but do you figure there might be some role for nutritional status?
Oh, I think absolutely, Lucilla. There's absolutely some. In fact, we have an exciting team grant with Janine Felix in Rotterdam at Erasmus and others to look at early life nutrition. I think all of these things play together. In many cases, they might have individual impacts, but they also probably have some combined and synergistic impacts. Of course, those are in human population studies not easy to untangle. But I think as we sort of learn more and have deeper phenotype cohorts and better characterization and you know a more sophisticated ways to analyze the epigenome, both on its own, but also in combination, as I very briefly touched upon, with the underpinning genome, we'll be able to decipher more carefully to what extent any of these environments, either individually or combined, contribute to the variability of the epigenome.
Yeah, I mean, the measurement of the metabolome in cord blood, for example, could be very useful.
We're doing this with the child cohort here in Canada that many of you will be familiar with, and other cohorts, and including microbiome, of course, is an equally exciting opportunity to bring in this sort of multi-layer omics to ask very similar questions.
Fantastic. Thank you very much. Thanks,
so
Dave.
Thanks, Lucilla. We have another question from Christian Mate. Congrats, Dr. Kabor. What kind of interventions do you think can change the fate of these children living in poverty during early life?
Again, a wonderful question. And you know, somebody a few years ago, I think it was actually maybe Judy Hall or John Charles, asked me what I think would be the next big question in epigenetics, and I said, well, one of them certainly is the impact of interventions. And so I wish I could tell you the data yet. We are actively looking at several nutritional interventions in conjunction with the project I mentioned with Dr. Felix in Rotterdam. We're also, as some of you know, we have been already publishing and now getting much deeper into the nurse family partnership intervention from David Olds that has a leg up here in Canada as well. And so I think if we again try to spread the umbrella wide to capture both interventions that might be of physical, nutritional, but also socio-emotional nature, we'll again get a better idea to what extent those might impact on the epigenome, and to what extent they might, if they do, those impacts might be distinct for each intervention, or there might be overlapping signals that we see. So right. Maybe in 2 years when I come back to a seminar, I'll show you some results on that.
Maybe I can ask one question from me. We've seen some really beautiful data recently from Archer Patronus and others that the process of methylation is very dynamic and it's changing constantly. And just wondering how you think that plays into the kind of signatures that you're looking at. And we think about it a lot. Lucilla talked about stress and glucocorticoids because we know that a lot of early environments can impact, glucocorticoid levels and regulation throughout life. And there's an ongoing conversation between glucocorticoids and methylation. So it's just how dynamic is the process? It's not just necessarily set in stone, right?
No, I don't think so. Although, as you probably know, when you follow some of the more critical voices in the field of epigenetic studies, they would believe that very little is actually changing. But then on the flip side, you take, the beautiful work that Art and others have done on particular on these sort of chronobiological associations with some of these methylation marks. So I think the jury is still out. We'll probably have to find some good middle ground between, very cell-based and more population-based studies, and certainly, accounting. I mean, there is this data, also beautiful data out from Rob Waterland and others, showing that there's a methylation signature depending on whether you were born or conceived in the dry season in Africa versus the rainy season and things like that. So I think there's much more to it. To what extent those are true epigenetic changes in the very same cell type, or as John Crelly would argue, perhaps some subtle shifts in cell type compositions that we don't necessarily pick up. I think that remains open, but I do agree that there is a certain dynamic in the epigenome, Steve.
I think probably we should wind things up. We're past the hour, and I would just like to thank Professor Kapoor for I think an outstanding lecture. It's certainly thought-provoking, and I think it's just fantastic, Mike, that you were able to take a break from your vacation to speak to us today. So thank you very much.
Great questions, and please be in touch, anyone who wants to discuss further, or again, as I said, I invite anyone who is interested in working with us or using the PET-BE clock. I think we're very excited about this, and if there are any trainees on the call, please also consider joining our team. We're always looking for wonderful and motivated and innovative and curious new people. So.
Thank you very much. That's a great offer for everybody. Thank you. Wonderful talk. I've really enjoyed it. Thank you so much.